concepts in biology are very much like a constantly changing world. After the discovery of the DNA double helix, significant efforts have focused on identifying genes and on understanding their contribution to human disease. Increasing numbers of genetic mutations and polymorphisms were shown to be important for the development of diseases, with as many as 40 different genes being sometimes linked to the same medical condition. However, for quite some time, multiple lines of evidence have indicated that genetic mutations are unable to explain by themselves phenotypic changes that shape disease. This means that additional factors which cannot be explained simply by changes in the DNA sequence have to be involved. In other words, gene expression can be shaped by mechanisms that do not alter the sequence of the nucleotides. This concept is supported by the high discordance rates between monozygotic twin pairs for many medical conditions, including certain diseases that historically have been considered to be highly heritable. Gene expression changes that occur without altering the sequence of nucleotides and are reversible and potentially heritable have become known as epigenetic changes. There are three types of epigenetic changes that have been described: the methylation of cytosines that are part of cytosine guanine dinucleotides, also known as CpG dinucleotides, the post-translational covalent modification of histones, and the microRNA-mediated negative regulation of gene expression at the post-transcriptional level. One of the distinguishing features of epigenetic changes is that, unlike genetic changes such as mutations, epigenetic changes are reversible. This opens the possibility to therapeutically modulate them and revert aberrant gene expression patterns to the pre-existing states, a finding that catalyzed the development of epigenetic therapies. As a result of these efforts, the first 4 epigenetic drugs were approved in recent years. 2 of them are DNA hypomethylating compounds, which means that they remove methyl groups from aberrantly methylated cytosines, and as a result, enable genes that became silenced by hypermethylation to become expressed again. 2 additional epigenetic drugs that inhibit histone deacetylases were approved, and they are very effective in treating hematopoietic malignancies. In my article, I am presenting some of the key concepts related to these 4 compounds, the promises that they offer for clinical medicine, and the challenges that are associated with their use. One of the major challenges is that these first-generation epigenetic drugs are somewhat promiscuous, as they can change the expression of many genes around the chromosome. This opens the need to develop more advanced second-generation compounds, which can selectively modulate the expression of a more limited number of genes. I am also discussing several compounds that are currently at various stages of preclinical and clinical development and emerge as promising epigenetic leads. One of the common denominators of these compounds is their promise to reshape virtually every medical field. Understanding the combined contribution of genetic and epigenetic factors to gene expression will be essential for dissecting the biological networks that shape development and disease, and for developing a new array of prophylactic, diagnostic, and therapeutic applications.